start with cerebellum and basal ganglia. So cerebellum, the word cerebellum, it has a bell. So I usually associate this bell with balance. So cerebellum has something to do with the balance. So if whenever you see some sort of balance problems, it usually leads to cerebellus issues. What about basal ganglia? Well, basal ganglia is just a bunch of neurons together and they perform some functions. We'll get them. So for basal ganglia, just remember this number called 5. It's 4 plus 1. I will tell you what. And in cerebellum, there is 5, 4, 3. 5 are the type of cells, 4 are the type of nuclei, 3 are the functions. In cerebellum, we have something it does with timing and coordination of the motor movement. Again, bell, balance. Well, cerebrum is totally different. Cerebellum is for the balance and coordination and timing. Okay. Histology: 5 cells, 4 nuclei, 3 functions. What are the 5 cells? 5 cells are 4 plus 1. It means 4 are same type of cells, 1 is the different. 4 same type of, 1 is the different. Okay. So in cerebellum we have 5 type of cells. The first cell I'm going to talk about is stellate, other is basket, Purkinje fibers. These three are, these stellate, basket, Purkinje fibers, and Golgi bodies. These are inhibitory cells, and these are 4. And the last plus 1 is granule cell. Okay, so let's talk about the names first. Stellate, it means they are star shaped. Basket is basket like network around Purkinje cells. So where are Purkinje cells? This is the Purkinje cells. Okay, so this is a Purkinje cell, and basket cell, it's covering this. It's basically a basket holding Purkinje cells. Okay. What are Purkinje cells? These are large flask. This is a flask. Okay, this is called a flask. A large flask shaped cell, large flask shaped neuron. Okay. Granule, these are positive, remember. Granule because they are granular, they are small grain, they are granular, small in size. What about dentate? Well, we have something called a dentate cells. We'll talk about dentate soon. This is dentate, and it's not cell, it's a nucleus. Tell you what. So these are 5 cells: first, second, third, fourth, and fifth. Note, all of them are inhibitory except granule. Granule is positive. It means the neurotransmitter is glutamate. And here, all of them, the neurotransmitter is GABA. Okay, so GABA and glutamate. Excitatory is glutamate, inhibitory is GABA. Cool. What about these layers? Well, there are 3 layers. First is the upper one, little bit middle one, and the lowest one. The external one is called external molecular layer. Cool, because it's externally there. The Purkinje cells have, what would they have? They would have Purkinje cells in it, of course. And the inner granular layer has both G in it. There is Golgi and granular. Okay, so how it works is that these cells are inhibitory. It means they are inhibiting something. What are they inhibiting? They are inhibiting either granule or they are inhibiting each other. We'll see about the circuits soon. It's very simple. It's not. It's very simple. Cool. So these are all the histology of. We have 3 layers: external Purkinje layer, inner granule layer. We have stellate cells, we have Purkinje cells, we have basket cells, we have Golgi cells, and we have a positive granule cells. GABA, and this is glutamate. Okay. Let's go into deep nuclei. Well, deep nuclei, it means they are deep embedded inside these layers, and all of them are excitatory, of course. Okay, and we have how many types? We have 4 type of nuclei. 5, 4, 3. We have 4 type of nuclei: dentate, we have emboliform, we have globus nucleus, and we have vestigial nucleus. Okay, so this is the medial side, this is the lateral side. Okay, so on the lateral we have something called dentate. The word dentate, it means dense, it's tooth-like. So this dentate is like tooth-like, you can see. Yeah, the form is tooth-like. So this is a largest nuclei, and it's really dense. Okay, what about other names? It's called vestigial. The word vestigial Vestigial is means it comes from the word vestigium. It means roof peak. Here's the roof, here's the peak. Roof peak, it's located at the midline of fourth ventricle. Remember this. So we know that there is lateral ventricles, and then we have this ventricle, and then we have, so basically you understand the, it is vestigial. It's present on the midline of the fourth ventricle. Okay, always remember this. Cool. What about other names? We have something called as emboliform. Well, the name emboliform comes from embolus, whatever you want to call it. And emboliform, it's plug adjacent to the dentate. It's an embolus adjacent to dentate. Okay, so this is like an embolus. You can see it's made like a drop of blood, or drop of, I don't know, green thing. So this is an emboliform nucleus. It's just adjacent to dentate. Dentate is the most lateral one, and vestigial is the most medial one. And the last one is globus nucleus. The word globus is, the word meaning of globus is sphere. Spherical. It appears round as cluster. As you can see, sphere, they are clusters. There are more like this, but we have only shown 2 of them, but there are more in numbers. So sphere, spherical, globus. So whenever you see globus, the word globe, it means sphere. Okay, so this is why we call them globus, because they are spherical in its nature. Emboliform, they are embolus. Dentate, tooth-like structure, largest, very dense. Vestigial, they are on the outside, they are on the medial fourth ventricle midline. Okay, this is the basic, definition of these nucleuses. Okay, so we know 5 cells, 4 nuclei, and 3 functions. We will tell about these 3 functions soon. The gross anatomy of cerebellum. So this is a cerebellum, this is a midline. Okay, so this is midline, and in the midline we have something called as vermis. So this is the vermis, right? This part is vermis. Okay, and this part is vermis. I will tell you why I have written this FGE, D and V. I will tell you why. Because vermis is the most functionally active part of the cerebellum, and it has 3 nucleus in it: F vestigial, G globus, and E emboliform. So these 3 are present here. Okay, of course vestigial has to be present here, because vestigial is present on the midline of 4th nucleus, so it has to be here. Okay, so vestigial is here. What about other zones? Well, we have intermediate zone. The intermediate zone is right here. Paravermis, intermediate zone. Okay, we call this zone as spinocerebellum, because they make this tract, because cerebellum, they give rise to fibers that go to spinal cord, and that's why we call them spinocerebellum. And because they are in the midline, of course, F vestigial also present at the midline of 4th ventricle. Because they are in the midline, they coordination of the motor movement of the trunk. Okay, the whole trunk of your body. Okay, what about the lateral hemisphere? Well, this is a lateral hemisphere. Okay, we can say this is left lateral, this is right lateral hemisphere, this is the lateral hemisphere, and we call this neocerebellum because it's newly formed. And the neocerebellum has something called as dentate nucleus, because dentate is present on the lateral side. Of course, vestigial is not going to be present on the lateral hemisphere, because vestigial is supposed to be present on the midline. Lateral dentate nucleus is present on the lateral, because it's present on the lateral. You can see this. So it's neocerebellum. It means it's newly formed. It's not highly. It's newly formed basically. It's neocerebellum. It's called neocerebellum. They're new. They are used for planning of motor functions. They are planning of motor function. And dentate planning means it has something to do. It has to be connected with cortical head of your brain, cortical portion of your brain. So it's neocerebellum. It's really fast, really vascularized, really myelinated. What about this part? Well, this part is called nodule. This is nodule because it's coming out, it's like a node. So nodule. And this is called a flocus. So the word flocus, it means flocus is a small tuft of wood. It's like a flocus. Okay, and we call it flocculonodular lobe, this lobe, and this is vestibulocerebellum because it's on the outside. Okay, and what else? It's an oldest part. So it means it's really less myelinated, or we can say it's really, the function is really primitive. That is equilibrium, the balance. Okay, and whenever you say vestibulo, it means it has something to do with ears and balance. So eye movement and balance, and it has a vestibular nuclei in it because it's called vestibulocerebellum. Okay, that's why we name them as they are. Okay, what about the physiology? Well, the physiology is that you have some sort of sensory input coming to them, and then the cerebellum calculates, the balance, or calculates the desired thing, and then cerebellum has motor, output. So this is the whole physiology of it. Now, how it works is that cerebellum has, start and stop function, and so there is first is start pathway, and then there is stop pathway. I will tell you what is starting. So what starts is this: that whenever you, for example, imagine you're standing, and okay, there's an example here. When archer pulls the string of a bow to release the arrow, so he's starting a movement, right? So this is a starting pathway. He's starting a movement. So start pathway is that it starts from inferior olivary bodies. So they are present, they look like olives. Okay, they are olives right in the medulla, and they are inferior because, for example, I make olive wait I make okay, so this is, for example, your medulla. So these are two olives right here. So they are inferior because they have to, produce fibers that go ascending. So inferior olivary bodies, they produce something called as climbing fibers. These ascending fibers climb the dendrite tree of Purkinje cells, right? So this dendritic tree of Purkinje cells. We know Purkinje are inhibitory. Okay, so climbing fibers, they go from olive to the deep cerebellar nuclei. Of course, these cerebellar nuclei is present here. Okay, so deep cerebellar nuclei, then it activates brainstem and thalamus, of course, and then it goes to cerebral cortex, and then the movement starts. Okay, and from cerebral cortex, the motor movement, spinocortical movements, spinocortical tract, and the movement starts. That's how the start pathway starts. So this is when you want to start any sort of movement. And when the archer releases his finger or the string due to inhibition of DCN. Okay, so this is the start pathway, this is the stop pathway. Why do we need stop pathway? We need stop pathway to calculate when the movement has to stop. Okay, for example, I tell you to pick up a pen. You will go and you will just move and pick the pen, and you will show me this, okay, I have picked the pen. But how this really happens, that your brain actually calculates the distance between you and the pen, and then you start your movement to calculate the pen, and then you move your hand a little bit towards the pen, and then cerebellum is constantly calculating the distance between you and pen. It's calculating the distance between you and the pen. As the distance is reducing, it keeps on getting reduced every second because you're going towards the pen. So at every minute second, the cerebellum is calculating this, the distance between you and the pen, and eventually you reach the pen, and then cerebellum says, okay, now you can pick it up. Just imagine your cerebellum was not working. If your cerebellum was not working, you wouldn't be able to calculate the distance between this pen and you would probably push your hand a little bit away from the pen, or a little bit short from the pen. I don't know how would it be, but you wouldn't be able to calculate the balance, between the motion. Okay, so this is the stop pathway. You stop. So again, inferior olivary nucleus, again climbing fibers, and then we have Purkinje cells, right? Purkinje cells present in the deep cerebral nuclei. Yeah, so these Purkinje cells are present in they inhibit deep cerebral nuclei. Of course, I told you these are inhibitory cells, right? and they inhibit these cells, right? And Purkinje cells inhibit deep cerebral nuclei and the movement stops. So basically it happens like this. What happens is that these climbing fibers, they can just either go directly to the deep cerebral nuclei to promote movement, and they can go to Purkinje cell to inhibit movement. Okay, so if these climbing fibers are going to Purkinje cells, they will inhibit the movement, they will stop the movement. If these climbing fibers are going to activate deep cerebral nuclei, they will promote the movement. So that's the stop pathway, that's the start pathway. Only difference is that climbing fibers will either go to deep cerebral nuclei, or they will either go to Purkinje cells. If they go to Purkinje, movement stops. Pre-start means you are calculating the distance, you're calculating the movement. Okay, so pre-start pathway. So pre-start pathway, it comes from spinal cord, of course, because your spinal cord is going to calculate. And this is a spinocerebellar tract. I just told you why. Here, here. Wait. This is a spinocerebellar tract. This part of your cerebellum calculates pre-start pathways. Okay, this vermis part, because this part is responsible for it. That's why we call it spinocerebellum. Okay, so this spinocerebellar tract. Okay, so dorsal cord, spinal cord gives fibers. We call them mossy fibers because they look like a moss. It's like a bunch of fibers like this, and they are mossy fibers. So they can either go to dorsally or they can go ventrally. If they go ventrally, they have to go through superior cerebellar peduncle. If they go dorsally, they have to go inferiorly. Why is that? If you check the anatomy of this cerebellum, you will see there are three peduncles. Here I will make three peduncles. Okay, so this is inferior, this is superior. Here it will be superior peduncle, that is ventrally. Here it will be inferior, that is dorsally. Okay, just remember this. Okay, so let's go to these mossy fibers. So mossy fibers via inferior cerebral peduncle dorsally go to granule cells, and via superior cerebral peduncle, that is ventral SCT, that is spinocerebellar tract, they go to granule cells. Now we know that all the cells in cerebellum are inhibitory except granule cells, and granule cells are positively, glutamately charged, so they activate these parallel fibers. All are positively. So if all are positively charged, it means they will work, they will start to work. And we know that all the cells in your cerebellum are inhibitory, it means they will stop. So what will happen is that granule cells will activate these cells, and these cells will inactivate the Purkinje cells. And we know that Purkinje cell is also for inhibition. So what happens is that granule cells hits these cells, basket cells, stellate cells, and Golgi inhibits Purkinje. So Purkinje work is to inhibit further movement. If Purkinje is inhibited, that means the movement will start, because Purkinje is responsible to stop the movement, right? We know that from start-stop signal pathway, right? So these cell, basket and stellate cells, they stop Purkinje, and Purkinje is stopped by climbing fibers. So I hope you get the whole story of it, right? So imagine if it was this thing was like if imagine there was no brakes on these, on this circuit. For example, granule cells keep hitting on basket, they keep hitting on stellate, they keep hitting on Golgi. What will happen is that eventually these cells will get out of control, and to have control over them, we have something called as feedback inhibition loop. So whenever that happens is that these granule cells, whenever they activate these cells, Golgi cells, the last G cell, because Golgi is present in the same layer, the same internal layer. Which layer? The same inner granule layer, right? Granule cells and Golgi cells are present together in a same layer. So Golgi cells can inhibit granule cells. That's why God has given Golgi cells and granule cells together. And that's why basket cells and stellate cells and Purkinje cells are close to each other, so that they can form a close communication with each other, right? I thought about it, like why God has not given Purkinje cells here or Golgi cells here, because it's hard to communicate far distances. So that's why Golgi cells are present in the same layer of granular cells. Okay, that's the whole concept of it. So Golgi cells also provide a negative feedback loop to granule cells, so that granule cells don't get overactive on these three cells, right? So this is a feedback because it's going back. What about this way? Well, this thing is a feed forward inhibition because they are inhibiting forwardly, right? They are not inhibiting backwardly. Golgi is not inhibiting forwardly. That's why it's called feedback. Okay, and feed forward inhibition is this. Okay, so Purkinje cells, deep cerebellar nuclei is inhibited. Again, we know deep cerebellar nuclei, we need this deep cerebellar nuclei for the movement. If this is inhibited somehow by Purkinje cells, the movement will stop. Okay, so its inhibition is lost. It means they are activated, movement starts. So that's why the whole circuitry is very simple. It's not complex at all. What about the archer? For example, the archer pulls the string again. The inhibition of the deep cerebral nuclei has to be removed, because deep cerebral nuclei is the nuclei which makes sure that the movement starts. So if you inhibit this deep cerebral nuclei, you will be inhibiting movement. So to inhibit, we need Purkinje cells, right? And if you want to inhibit Purkinje cells, we need other three cells, that is basket, stellate, Golgi. And to inhibit granular, we need Golgi. So this is the whole story of it, okay? I hope you really understand this because it's really, really, really simple. Okay, what are the signs of cerebral lesions? Okay, what are the signs? Well, the first sign is truncus ataxia. Well, truncal ataxia, it means the vermix controls the trunk. I told you this is cerebellum, this is the vermis, this is paravermis, this is lateral hemisphere, this is the nodule, this is floccular part. Okay, so this part, vermis, controls the trunk of your body, the whole trunk. Okay, so vermis controls the trunk. If lesion is there present at the vermis, there will be bad posture, right? If there is any sign of lesion on cerebellum, there will be no start stop signaling, because we know this is the whole function, this is the function. I told you there are three functions, right? This is the three functions. Okay, start pathway, stop pathway, and pre-start pathway. This is the three function of cerebellum, right? Okay, what about wide base stance? Well, a person will have wide base stance because the vermis is damaged and he's trying to not fall, because cerebellum keeps calculating your posture, right? If your posture is bad, you will fall either ahead or backwards, right? So you will have wide base stance, you will stand your legs apart so that you don't fall. What about titubation? Well, titubation is tremor of the head. Well, if there is lesion at the vermis, neck head balance will be messed up. The cerebellum corrects this balance all the time. Okay, it's like calculating in real time. Okay, it calculates every minute detail. What about ataxic drunken gait? Well, drunken gait, always remember, drunken gait is because of cerebellum, because drunken gait is when vermis and paravermis is damaged, and then you have messed up gait. Okay, because you can't balance yourself. So messed up, it happens because of alcohol suppress Purkinje fibers. Okay, because alcohol, we know that Purkinje fibers, they inhibit this deep cellular nucleus. Okay, and if this is inhibited, the movement stops. Just imagine your Purkinje cells are always suppressed. It means this is always upregulated. If this cerebral nucleus is always upregulated, it means the movement never stops. You will keep moving. You will keep, moving like a drunken guy. So alcohol, it messes up with the Purkinje cells. What about nystagmus? Well, nystagmus is like when your eyes are going like this. Okay, when your eye movement is going like this, left, right, left, right. Okay, so nystagmus. Well, floccular nodular lesion, because eye is involved. Floccular nodular, loss of smooth pursuit. Smooth pursuit of what? Smooth pursuit of balance. Okay, loss of smooth pursuit, eye drift off. Because imagine you're on a ship. Okay, and the ship keep getting left, right, left, right, right. So you have to balance yourself. So that's why you will have this nystagmus, because you're trying to balance on a ship and in reality you're just standing on the ground, but your body thinks that you're on a, you're on a plank, a wooden plank, and this plank is on a triangle, and you're standing on it, right? These are your legs, this is your body, this is your head, this is your hands, and you're like balancing yourself. So nystagmus is there. What about decomposition of movement? Well, decomposition of movement, it means it will not be a very smooth movement. Okay, it will be no fluid movement. Why there will be no fluid movement? Because the smoothness comes from pre-start calculations. Okay, whenever you are able to calculate the whenever you're able to pre-calculate any sort of movement, the smoothness comes. Just imagine your pre-start pathway is messed up. If pre-start pathway is messed up, of course your decomposition of movement will be there. What about pass pointing? Well, finger nose test. You have to start uh, touching your nose from your hand distance to your nose, and you won't be able to calculate this distance because your cerebellum is messed up. Okay. What about kinetic intentional kinetic intentional tremors seen with activity absent at rest? Okay, so the more you move, the more tremors starts because inhibition is lost in cerebellum. That's why. What about dysarthria? Dysarthria means scanning speech. Laryngeal and tongue muscles are also controlled by cerebellum, and if cerebellum is messed up, your laryngeal and tongue muscles are messed up, you won't be able to speak fluidly. You will speak like I am, like this, scanning speech. Okay, so what about peduncular knee jerk? Well, cerebellum normally modulates tone and reflex. So imagine if your cerebellum is messed up, you will have something called as peduncular knee jerk. What about dysdiadochonesis? Dysdiaconesis. What is diadochonesis? Wow, weird name, a long name. So the meaning is that inability to perform rapid switching, inability to perform alternating supination and pronation. Okay, you won't be able to do pronation, and you won't be able to do supination, because both of these movement, pronation and supination, are done by switching between agonist muscles and antagonist muscles. Okay, so this switching is done by cerebellum. If your cerebellum is messed up, you won't be able to do this switching. What about rebound phenomenon? Unable to stop the movement. If you keep on, if you just keep walking, you won't be able to stop. Yeah, right. Ask a patient who is suffering from cerebellum lesion. Ask a patient to walk. If he starts walking and ask them to stop suddenly, he won't be able to stop suddenly because this inhibition, the check reflex, is gone. He won't be able to inhibit because the Purkinje fibers are messed up, so the movement never stops. That's the whole concept of cerebellum movements. Okay, I hope this topic is really clear. The cerebellum.